Good afternoon one and all. This is Praneeth and I am going to teach you a class about the cell, which is the unit and structural unit of life. Before coming to cell, what is a cell? You may hear many things about a cell. For example, if you take what is the meaning of cell in biology, it is the structural and basic unit of life. And if you go to a physics or physical science, the cell is defined as the electrical unit which gives power or electrical source to the equipment. And if it comes to some local terms like cell, basically we use it as mobile phone, and otherwise you call cell in computers. It comprises of a box which is made up of the 4 borders or comprises of 4 compartments, is called a cell. Now coming to biology, what is a cell and what are it? Everything we know. To make a house, we need building blocks or bricks. By arranging one brick over other, we used to build a house. The key component, obviously the smaller component will be the brick. And other inside our body is made up of tissues, organs, and coming to the organic level, ourselves we call it as organism. From organism, the lower level we call it as organs, and the organs is made up of tissues, and the tissues is made up of cells. According to coming to the discovery of the cell, you all may know because in the previous years like 19th century and 18th century and before that, we don't have microscopes, isn't it? Because since cell is a very tiny little structure, we can't see them through the naked eye. Means with our eye, we can't see the minute particles and the particles which are less than approximately 20 microns. After the invention of the microscope, Robert Leeuwenhoek, in the year 1665, he discovered cell. Means he took out a cork. In general, we know very well cork in the example of it is used in the bottle caps, like glass bottle caps. He took out this cork stem and he cutted that cork stem transversely. Transverse means if we take a stem like this and if we cut along perpendicular to the axis of the center, which is called transverse. He took a transverse and he observed it under the microscope. After the observation, what he used to see is the tissue is made up of look like a box, not a box, but a closed chambers like structures, in which he named it as cell. Suppose to our consideration and to our understanding, we can know simple cells like in your house you may take onions and take an onion and you may observe, first peel off the cover and slice it nicely. Take a small piece and open it. If you are taking a piece like this and cut it or fold it into twice, means if this is a one onion piece, fold it into twice, then open it and you will have a semi-transparent film like this. I will show you. If you are taking an onion, this side will be the outer part and this side will be the inner part, and if you take an onion like this, if you fold it and you open it like this, you will have a semi-transparent film which is called onion peel. And take out that onion peel, and place it on the glass slide. Usually these slides are made up of glass and these are useful to take observation in the microscope. This glass, take that onion peel and place it on the slide. Mount with the solution and place a cover slip on it and observe it under the microscope. You will see the cells of the onion peel, which will have walls around it and the center thickened part, which is called it as nucleus. And you can also do this activity by taking the human cheek cells. For this experiment, I will give you a small demonstration. Take out a small spoon or small toothpick and gargle your mouth with water, clean yourself and take out the pick and rub along the sides of the cheek. And you can make it and drop this cheek and the cheek cells on the slide and put it on the slide, and after adding stain, make sure you add glycerin for protection and paste a cover slip for, and you keep the slide down the microscope and you observe these things. How are the cheek cells and how are the onion peel cells different from each other? And after that, coming to, we know we have many plants around us, and coming to the plant cells, there are two kinds of cells obviously. The one is the plants and another one is the animals. Plant cells and animal cells are completely different because plant cells synthesize their own food because they are autotrophs. They do photosynthesis they will have chlorophyll pigment. But in our animal cells, most of the cells may not have chlorophyll because it depends upon another organism or other food. Now coming to cell, since it is a structural and basic functional unit of any living organism, cell is the most important vital organism, vital part of the body which coordinates each and every other cell and which will make up of a tissue, and the tissues will make up of into an organ, and the organ is modified into organ systems and organ systems to human organism, not only human but an organism. I will give you a small example. And let us take a small example of a liver cell. If the liver cells will be approximately the size of microns to 20 microns to 80 microns. Microns is a unit of microscopic size of a cell. Microns is denoted by symbol mu, and after that I am taking a group of liver cells, and the liver cells they accumulate and made up of a organ into liver, and the liver then it will be made up into an organ system. Organ system means which we construct our digestive system, and after the digestive system include the digestive system, our respiratory system, our nervous system, and all the organ systems combinedly made up and forms an organism. And after that simple example I would like to tell you, take out a small leaf, and we all may know taking leaves, any kind of leaf. For your best example, your available leaves will be right kind of hibiscus and rose. Many of you will get right because these are the most common plants in our household and all. Take out a small leaf, observe the the leaf have two surfaces. One is on the top side and the other one is the back side. Observe any other side, but I would like to suggest, because the lower side will have many stomata. Stomata means you I think you guys know, because stomata is the opening of the leaves which exchange of gases occur through. Then of observing the epidermal layer of the back side of the leaf, you would notice stomata and normal cells, means epidermis cells and the stomata. You can differentiate because the stomatal cells are like kidney shaped cells which will be arranged in like this shape. Two will be there, and the outer inner exact middle position will be the opening of the stomata, and you can notice it through an ordinary microscope or with your hand lens, means magnifying lens. You can notice it very well, the structure and shape of the cells. Coming to plant cells, you can how you can differentiate and how you can take out the plant cells. If I want to take out how the epidermis is adjusted on the leaf, I would like to take a blueprint or copy of the epidermis. With the example, I would like to take out a leaf and paint a nail polish. Many of you guys will know the nail polish, and apply 1 or 2 coats which will be light colored. I would like to use sky blue or violet or some pinkish red. That colors will be nice, and apply one thin coat, and after the drying apply another thin coat, and after applying take out the layer of the nail polish from the leaf and place it on the slide and observe it under the microscope, and you will get very nice display of the cells of the epidermis of the leaf. And not on, I would also suggest you do with other leaves also, because not every leaf may be differentiated, because one leaf will be like if you take a example between the hibiscus leaf and coconut leaf, the epidermis and all structures and the stomatal will be different from each other because each plant have its own modification. And I will also suggest you do your activities like observe your leaves or observe your and I would like also suggest you to observe the different kinds of cells which will use the which you guys will know, because taking out the plants and taking out the fruits epidermis layer and take out of different ones and observe it under the microscope or simple magnifying glass, you will get a different and wide variety of structures and wide variety of shapes also. And now I would like to tell you about how to do or how to take and how to observe the plant cells. Before coming to that, of course we have to place it on a microscope. By placing on a microscope, we can't keep the whole plant or whole thing on the microscope and we can't see. For that we used to make a small sections means a thin part which we used to cut, a small sections in which we used to keep it under the microscope. There are two types of sections primarily. One is longitudinal and another one is transversal. Means you guys will know that the LS of a flower and TS of a flower or TS of a stem. For the LS means longitudinal, means vertical, which you guys know vertical means obviously straight from up to down. Means if you are taking a stem, you should cut it into vertical, or if you are taking a flower, you should cut it into vertical. That is called longitudinal section. Of maximum, I used to suggest you longitudinal section is used for only to study the morphology, means how the systems are arranged or how the plant parts are arranged or how the floral parts are arranged. But for the study of the internal cells and the arrangement of cells, I would like to suggest you to take out transverse cutting. Transverse cutting is simple, guys. It is to the plant axis, means if the plant is like this, or the stem is like this, take out a perpendicular stretch. Means if it is like this, I would like to cut it along perpendicular axis. Means you will get, for suppose, if you take this as stem. If I want to take is a transverse section, I would like to cut it along this line, because for your differentiation I would like this is one total part, and after cutting this will be the top part and this will be the down part. It will be like this. Means we should observe like this. Means we will get a round shape because it is cut into transverse, and after that we can take it a smaller sections by cutting like this. We are taking the smaller cutting, which is also a transverse sections. By taking the transverse sections, we are taking very small thin films, because if you are getting the thicker, the more cells we will get, and you can't able to differentiate what are the cells or what are the shape you are taking, and we can't differentiate the nucleus in the cells. Take out a small layer and place it on the slide, and if some plant cells are like this, they will be transparent little. Then we would like to add some stains. I'll give you example of stains, the saffronin and methylene blue and cotton blue and all these things. These stains are used to just to thicken the walls, of which it will fill a color for our coloration, means to visible. Means we can't observe the thickness and all, the outer cell walls and all, because some may be the one layer of the cell may be overlapped by another layer. To get differentiated, we get used to stain. And after that placing pour 1 or 2 drops of glycerin. The glycerin is used not to dry up the cells which we are taking. Since we are taking approximately 2 to 3 layers of cells, if we are keeping it on the slide for a long time, the cells would be dried up. After drying, we can't distinguish the structure and shape of the cell, because the water content in the cells may be evaporated. To prevent the evaporation, we are using glycerin. After using the glycerin, we are making another cover slip, which will be a thin glass, to mount it on. By placing the cover slip, it would not be moving to left or right, and it would be in a firm position. And after that placing, we are taking our plant TS and checking out under the microscope. You will get the outer one as epidermis layer and another one cortex and all. Into the centermost part you will get medulla and xylem and phloem and all these things which you will get in the study higher classes. And I would also like to share the types of cells, like you all guys know that there are different types of cells which will be of different shape. And of course the nerve cells will be of a one head like, and it will have the branches and all, and one long middle sheath, the neck and all, and after that it will have dendrites, that it looks like almost a tree. And for the smooth muscle cells, it would have a spindle like shape. Like this shape it will be there. And also if you are taking the cardiac cells, means which is the heart muscle cells, it will be of different constraints and it will be interlinked to one another. And of course I would like to suggest the activity of the blood cells. You guys all know what is the blood. Blood is a fluid component tissue, connective tissue of every living organism. Okay, while observing blood cells, you all know the three type of blood cells. One is the red blood cells, and another one is the white blood cells, and another one is the blood platelets. And of course red blood cells will be of circular shape, which will be a biconcave, like you all guys know the structure of like Gary, the thing which you used to eat without a hole. It will be look like as same the red blood cells. It's the shape, like it will be a round, and the both the sides will be concave. And after the white blood cells, they are irregular in shape. They have nucleus also. The nucleus, they will have the nucleus to one or two lobes. Most of the living cells will have single nucleus, and in some conditions it will have binucleus or two or more than other nucleus. And after that the nucleus will be either spherical or lobed. Lobed means which will be divided into lobes. In the red blood cells you won't get notice any nucleus, because red blood cells is enucleated, means in the red blood cells the nucleus are absent. And after that the white blood cells, you will have leukocytes, basophil, monophil, acidophil, eosinophil and neutrophil. These are the 5 types of white blood cells you have. And after that they will have the different lobes and configurations, and all the sizes also it will be vary. And the last one, the blood platelets, it will be of it is also irregular of shape. It plays a role in the blood clotting. And after that I would like you guys to suggest observe each and other different cells which are in our human body. I will give you an example. First observe the skin cells, which is the outermost layer, skin cells. And after that, moving to the internal, observe the fat cells in our body. Fat cells will be lipids or fat storaging cells which will be underneath the skin. After outermost layer will be epidermis for all, and the inner side you will have fat cells. Fat cells will be different because it have to store the fat. And after that inner, we can go either the muscle cells. Muscle cells, you guys will know the muscle cells will be long, straight and spindle shape. It will be interconnecting the bones or the tendons. And the muscle cells, after observe the cells which are lining up the what we call blood vessels. The blood vessel cells are somewhat cylindrical and some are like plate-like structures, in which they made up of a cylindrical like structures which they are used to transport the blood and all things. You guys will know that different cells and different compositions, and the shapes also will be different from each other. Some will be like cuboidal structures like epithelial cells, and some will be of round cells like which will be of in our glands, and some cells are like long and some net-like structures which you will see in the nerve cells. And you also try to identify the bone cells. Bone cells is one of the most complicated cells because its structure is somewhat different from every other cell, because it is a somewhat solid-like structure. And hope you guys understand this lesson well. And on the finishing, I'm taking this lesson to St.Josephs School, Vellachintalagudem, because Sister Janet has advised me to take a class about this. Just it is my small video and small contribution from myself to sister, and I am also a student of St.Josephs school. Thank you guys for listening, and if you guys like this, please support me. Thank you.